boy, this would be a hard slide to have to memorize. So now we've got our cells from however we got them, from an embryo, from an adult, from switching the genes. Now we've got them, what are we going to do? We're going to direct the differentiation. So over here, this shows we have some human embryonic stem cells. We start adding different things to this dish. And depending on what you add, you get different things. So this is, we got a neuron, Schwann cells, fat cells, cartilage cells, bone cells, smooth muscle cells. So, and this takes a lot of work. It's a lot of guess. It's a lot of trial and error. So we've got them, now we can do this. This is just an example with the bone marrow cell, quickly so that you can see this hematopoietic stem cell that we get from our bone marrow. We can direct to form any type of blood cell we need, plus other types of cells. That's just an example, real quick. And there it is. It looks easy on paper or in pixels. I guess we don't call this paper anymore. Okay. So now we're going to jump to the potential uses. So now we can make bone, and we can make cartilage, and we can make fat. Don't know why. We can make all sorts of things. So what are we going to do with that? One great thing to do would be to test drugs. So we use animal models a lot, but wouldn't it be nice if we had cells right there, if we had neurons that were just like ours, that we could test and see if the drug would work or if it was toxic or whatever. We can use them for drug production. So like I said, those bacteria, we gave a little bit of DNA and they produced insulin. Well, we can take some of our own cells and cause them to make insulin, so it would really be human insulin from human cells. Okay? We can study how disease progresses. So if we made those cells specifically with the disease, we allow them to grow and we can see what different things are happening as the disease moves along. One great thing that happened when we were able to get embryonic stem cells, human or other animal, was that we started to understand how human embryonic development proceeds. And that could help us with understanding disease as well. And then, of course, what I've been talking about is regeneration and tissue engineering. So let's make a heart, let's make a liver, let's make a new piece of cartilage for your knee. So that's where it would be nice to have whole organs for transplant. This is a picture, and I put where that's from. This is from Cord Blood Bank, one of the cord blood banks in the U.S. So anyway, this just shows the different kinds of diseases we are hoping to be able to cure. So if you have a stroke, you have a block in blood vessels. If you can make new blood vessels, allow those to grow in the brain, that would be really useful. Okay, so if we had heart disease and we can repair the heart with stem cells, that would be great. Diabetes I've talked about. Parkinson's and Alzheimer's are problems in the brain, so we could put some fresh cells in there. That'd be awesome too. So you can read through this. I'll tell you how to get to these slides later. Oops. How many people have had a transfusion in here? I'm the only one who's had a transfusion in this whole room. Okay, my mom's had like 5 or 6, so she'll make up for the fact that you guys haven't. The thing about transfusions is we tend to have a little, a shortage of blood. They're always sending the bloodmobile around saying we need blood. So there's a shortage. And also disease could be in there. So after I had my transfusion, I'm like, oh, I know they say they check for this, and that, but I hope nothing shows up later. So that's an issue. But wouldn't it be nice if we can make this? We just had reported in July, August, real recently, that this particular group made red blood cells. And this was hard to do, but they were able to do it. So now they just need to figure out, can they do this on a large scale? Can they make the O and O positive and the B and the A? Can they make all of them and do it at a large scale? And can they do that cost effectively, cheaper than collecting blood from other people? So it was a difficult thing. Spinal cord repair. So as you know, Christopher Reeve, he has, now that he's dead, there is a foundation for the study of how spinal cord repair can happen. His brother Brock Reeve is really big. His name's everywhere. And then, so what they think is if they add different stem cells to the injury, we have a couple things going on. One is it might be a bridge, actual mechanical bridge, or maybe a chemical cue. And then, so there's lots of other things that's happening. So I'm not going to read the whole slide. Diabetes I've talked about, again, so we could talk about this. This was from bone marrow. The green cells are from bone marrow. The red ones are that they actually became cells that can secrete insulin. Just so you know, countries like China, Ukraine, Russia, and Brazil, they all have like these stem cell clinics. Our FDA will not allow this. So there's a lot of sort of so-called testing, anecdotal testing going on to cure heart disease. So I'm sliding through these pretty quick because Dave is standing.
Yeah, Dave.
These people made heart valves. They actually collected amniotic fluid, got the cells, and they made heart valves for replacement. So if you ever have a mitral valve prolapse. So I'm just pointing out this last slide because I used a lot of the internet, but I do want to point out there are 3 really good books. So if you can get those in the bookstore, the Stem Cell Dilemma is the newest. The one I really, really like is Stem Cell Now. The Stem Cell Divide is good too. A lot of people send me emails saying, oh, where's a clinical trial? I don't know, but there is a website called clinicaltrials.gov. So if you're interested in a stem cell trial, you can join up there. Again, there's the stem cells at the NIH site. And then my website, I have a personal website, I have a couple course websites, but my personal website is called JoanneLovesScience.com. And there, if you have a question about stem cells, you can contact me through there. You can also find this presentation. So if you ever want to refer back, it will be there. And I don't know, maybe Dave will put it on the Parkland site or something, so we could do that. So since we're wrapping things up, I just want to say thank you. Do we have time for like 2 questions?
Yeah, very little, but we maybe want to take a couple of questions. Any questions for Joanne?